everything, of course, makes more sense with an example. So let's work through a couple of examples so we can better understand how we do phylogenetics. Phylogenetics is a lot of fun, so let's work through some problems. So here, we're going to start with a relatively simple problem. So we might start with a table. This is a table that looks something like this. You can see on the left-hand column, we have our characters. So we're looking at the nose, tooth comb, postorbital bar or postorbital closure, and the type of vision. And then along the top, you can see we have a couple different species here. We have a tree shrew, a lemur, a tarsier, macaque, and human. Right now, we're not being super specific. Of course, if you want to do a real one, you want to, be like, oh, this is Macaca nigra, this is Tarsius tumpara, and be really specific about what species you're including. So here, these are the characters or traits. Those terms are pretty much interchangeable. Over here, now we have what are called the character states. It's what type of trait do we have? So you see, for the nose, we either have a rhinarium or we don't have a rhinarium. For the tooth comb, you either have one or you don't. But it might also be different types of things. So look at the bottom for vision, they're either dichromatic or trichromatic. So it really depends on what type of trait you're talking about, how you code that polarity. Another important thing to note is over here we have our tree shrew or our outgroup. So we know tree shrews are not primates, so we use those as our outgroup to compare different primates to each other. The next part on character polarity is we want to code it and figure out what is primitive or simplesiomorphic and what is derived or synapomorphic. Here I've used the colors gray and green to denote them. So anything that matches the tree shrew, we're assuming that is primitive. And that's a pretty common assumption that people make. We use our outgroup as our assumption of what the primitive or simplesiomorphic state is. That's not always true though, because remember the tree shrew has been evolving for just as long as we are, so it might also just be derived in a different direction. And here, anything that does not match our tree shrew, we are assuming it is derived. Of course, we never know if that's quite what's going on, but that is a decent assumption to start with. So based off of this, relatively simplistic character tree, we can end up with this type of tree here. So we have our tree shrew. We know that's our outgroup, so we use that to root our tree, and everything else is more closely related to each other. You can see the human and macaque of the traits that we looked at, they have exactly the same state for all of them. So we know they are more closely related to each other. And then we see the tarsier shares a little bit more with the human and macaque. So we link that with the macaque and human. And then our lemur outside of that. And of course, our tree shrew on the very outside. When I'm doing things by hand like this, I tend to try and link things that are more closely related to each other first. And then I add everything successively from there. Of course, we don't do it by hand anymore. Nobody actually does it by hand. It's helpful to do it by hand when you're just starting out so you can understand the principles, but of course, now we have lots more data. What people are doing now is we sequence lots of DNA, and I do mean lots. Sometimes this does mean full genomes, but frequently when we're doing phylogenetics, we actually don't have enough money to do full genomes for multiple individuals, because that's what you need if you're doing phylogenetics. You need multiple individuals to really connect them and see how different species are related to each other. So for that, people tend to do a method where you get a reduced subset of the full genome. Some common methods I've seen are you can sequence exomes or all of the genes that are expressed in a genome. Though what is more common for phylogenetics right now that I'm seeing a lot of is variations on RAD-seq. So RAD-seq is restriction-associated DNA. So you have a genome, you throw a restriction enzyme at it, and just by chance it gets, like 10,000 different spots in the genome. So that's a good way to get, many different spots in the genome. Like 10,000 is a big number. But it's still less than a full genome, so it's not quite as expensive. So this is a method to try to get good coverage in many different places in the genome without breaking the bank. The next part we do computer magic. Thank God for the field of bioinformatics, because this field has been focusing on how to align sequences, because once you sequence your DNA, first you need to align and make sure you have the same place and that they're all matched up so you can interpret it. And then once you have them aligned, you interpret relationships. Thankfully, the computer does all of this for us. but really it's a bunch of really wonderful people who have worked on creating the algorithms so that the computer can do it for us. But let's talk about one more example. My favorite, tarsiers, and they are in fact the problem. Let's go back. So for many decades, there's been a debate on how we should classify primates and what taxonomic groupings we should use. Today, we have pretty strong support for strepsirrhines versus haplorhines, and that's what I've been talking about in this class. However, some people do like to say that we should instead use prosimians and anthropoids to classify primates. And really, tarsiers are the pivotal group. And where do they go? So if we group tarsiers with anthropoids, that's when we get this name haplorhines. If we group them with strepsirrhines or lemurs and lorises, that's where we get this term prosimians. So let's talk about why we have this debate in the first place. So first, tarsiers do have a few primitive traits. So, they have a grooming claw. They have this leaping locomotion. They have an unfused mandibular symphysis. So this means the two halves of their mandible, they actually aren't connected by bone in the center. It's connected with cartilage, so it's not like completely disconnected, but it's, a little bit of flexibility there. Our jaws are fused. They also have simple teeth, sharp, tiny, pointy teeth of death here. And they have a bicornate uterus. This is a primitive mammalian trait. So instead of like being a single chambered uterus, it's actually kind of this Y shape with two horns. That's where the bicornate comes in. And they also have multiple nipples, another primitive mammalian trait. Tarsiers do have some derived traits, so they do not have a rhinarium like us. They do have a few derived traits in their eyes, such as a fovea, macula lutea, high central cone density or high cone density in the central retina. They also don't have the tapetum lucidum. So these are derived and do not match them with lemurs and lorises. However, we have a third category. They have a bunch of unique traits. So these are their autapomorphies. So they are entirely carnivorous, and they're the only primate that's entirely carnivorous. They also have these super elongated tarsal bones in their ankles. So they elongated their ankle bones to make their legs longer so they can jump even more. And some of them can communicate in the ultrasound, which is super cool. But there's more. They also have these extreme orbital hypertrophy. Like, their eyes are so big, we needed to use two words to mean big, because extreme and hypertrophy. And they also have this partial orbital closure. So you can see in the back of their orbits here, they have a little bit of that closure, but not all the way. So it's unclear, like, did they try to have the complete orbital closure like we do and other anthropoids? Or, and is this just, their eyes are so big, they were unable to do it? So that would suggest homology. Or is this just completely different and doing it its own thing? People are still arguing about this. We can also look at the three different tarsier groups here. So we have the Philippine tarsier, Tarsius. We have the eastern, Carlito. We have the eastern tarsier, Tarsius, and then the western tarsier, Cephalopicus. We think we have a decent idea of how these three genera are related to each other. But the Tarsius here has a normal number of chromosomes of 46. And these two guys have 80. That's a weird number. That's very high for a mammal, and we don't know what's going on. But let's talk about what this means. So we can put all these traits on one page. So we have simplesiomorphies, which link them to strepsirrhines. We have synapomorphies, which link them with anthropoids. Then we have these autapomorphies, these just bizarre characters that we don't know what's going on. But having three different groups of traits like this means we have big difficulties with Tarsier phylogenetics, and how do we classify them? So these primitive traits link them to lemurs and lorises. Their derived traits link them to anthropoids, and their unique traits suggest that they're just maybe they're different than everyone. So this means different people have come up with hypotheses for how we think tarsiers are related to primates. You might get this tree. So here, tarsiers are related to lemurs and lorises. So in this tree, we are emphasizing these simplesiomorphic characters and how different anthropoids are. Here's another tree. So now tarsiers are just separate from everyone. So here we're emphasizing the bizarre traits we see in tarsiers, and they are a separate lineage. And now we have a third tree, and here we're grouping them according to synapomorphies. Of course, these two trees at the top, we do not think this is what's going on, and we think this tree at the bottom where we are grouping by synapomorphies is the true tree, But because there are so many different lines of evidence that suggest different things, it's taken a long time to figure out exactly where tarsiers fit. Even when we're looking at DNA, some of the early genetic analyses put tarsiers outside of primates entirely. And it's only with more robust genetic data are we actually seeing a well-resolved tree like this at the bottom. So, how do we use phylogenetics? How can we infer evolutionary history most confidently? But also, how do tarsiers cause problems when inferring primate phylogenies?